Hello, my name is Professor Hudson. Let's continue with the narrated PowerPoint lectures for Chapter 9, covering mitotic cell division and cancer. Let's take a look at mitosis in more detail. As mentioned previously, mitosis is the part of the eukaryotic cell divisional cycle where the chromosomes that were duplicated during Anaphase are actually separated. Mitotic cell division consists of nuclear division and cytoplasmic division. When mitosis is initiated, each chromosome has two sister chromatids attached at the centromere. Mitosis has four main phases based on appearance and behavior of the chromosomes: prophase, metaphase, anaphase, and telophase. This figure shows an animal cell in mitosis. It is showing what the chromosomes are doing at each stage of mitosis. Note that some textbooks will include another stage called prometaphase. Prometaphase occurs at the end of prophase and the beginning of metaphase. The first phase of mitosis is prophase. 5 major events occur during prophase. Duplicated chromosomes condense, spindle microtubules form, the nuclear envelope breaks down. Chromosome condensation causes the nucleolus to break down. Chromosomes are captured by the spindle microtubules organized by centrioles. So during early prophase, the nucleus and the nucleolus must disappear, and the spindle fibers much attached to the chromosomes. Late prophase, which is sometimes referred to as prometaphase, has the following highlights. The chromosomes continue to condense, the spindle microtubules form, the nuclear envelope breaks down, and the chromosomes are captured by the spindle microtubules. This is happening during early prophase and continues during late prophase. 2 types of spindle microtubules form: polar microtubules and kinetochore microtubules. The spindle microtubules radiate from the poles, both toward the nucleus, forming a basket around and outward towards the plasma membrane. The nuclear envelope disintegrates, releasing the duplicated chromosomes. The kinetochore is a protein-containing structure located at the centromere that will attach to the spindle fibers. The spindle is organized by the centrioles. Plants, fungi, and some algae lack centrioles, but they still form spindle poles. During metaphase, the chromosomes line up along the middle of the cell, sometimes referred to as the equator, the metaphase plate, or the equatorial plane. When the chromosomes are lined up single file in the middle of the cell, we say the cell is in metaphase. The kinetochores of each duplicated chromosome are connected to the spindle microtubules, leading to opposite poles of the cell. The spindle fibers are what pull the chromosomes into the middle of the cell. During anaphase, the sister chromatids separate and are pulled to opposite poles of the cell. When you see the chromosomes separate in half, with the chromatids being pulled apart towards the opposite ends of the cell, we say the cell is in anaphase. Motor proteins in kinetochore pull chromatids apart along the kinetochore microtubules and toward the opposite poles. At about the same time, polar microtubules form opposite poles attached to one another where they overlap at the equator. These polar microtubules then simultaneously lengthen and push on one another, which forces the poles of the cell apart so that the cell assumes an oval shape. So the microtubules, which make up the spindle helps to pull the chromosomes apart, or I should say the chromatids of the chromosomes apart, and the spindle also helps to elongate the cell. We've entered telophase when you see the nuclear envelope reforming around each group of chromosomes. Telophase is the end stage of mitotic cell division. The spindle microtubule disintegrates. A nuclear membrane forms around each group of chromosomes at the pole. Chromosomes unwind and revert to their extended state. The nucleoli reappears. Cytokinesis can be seen as well. During cytokinesis, the cytoplasm is divided between two daughter cells. This slide and the one that follows summarizes mitotic cell cycle with cytokinesis. It shows a cartoon of each stage along with fluorescent microscope pictures showing an actual cell going through each stage. Please make sure you know each substage of interphase and each stage of mitosis, along with key things that happen in each stage. Remember, interphase consists of G1, S, and G2, and then mitosis consists of prophase, which is sometimes divided into early and late, with late prophase being called prometaphase, metaphase, anaphase, telophase, and cytokinesis. Let's talk about cytokinesis. Cytokinesis is division of the cytoplasm in organelles. Cytokinesis differs greatly between animal cells and plant cells. In animal cells, microfilaments attach to the plasma membrane and form a ring around the equator of a cell. The ring contracts and constricts the cell's equator. Eventually, contraction of the ring pinches off the membrane, forming two daughter cells, each with a nucleus identical with the other. When the ring contracts and constricts the cell at the equator, we sometimes refer to this as the cleavage furrow. Following cytokinesis, animal cells enter G1 of interphase, and depending on the cell type, can start the whole process again. In plant cells, you will not see the cleavage furrow. Instead, a cell plate will form, which will eventually give rise to the cell wall. This slide is illustrating what happens with cytokinesis in animal cells. Note the contractile ring which pulls the cell into two cells, and we refer to this region as the cleavage furrow. In plant cells, as mentioned previously, there is no cleavage furrow. Instead, vesicles begin to migrate to the middle of the cell to form a cell plate, and then eventually this cell plate will become a cell wall. So in plants, the stiff plant cell wall prevents the pinching off of cytokinesis seen in animal cells. So instead, these carbohydrate-filled vesicles will form the cell plate, which will give rise to the cell wall. Here's an electron microscope picture showing the cell plate forming between two cells that have divided through mitotic cell division and cytokinesis in a plant. Let's review. Question 6. Which of the following is not associated with prophase? Choose the correct answer. After blank, nuclear division is completed. Choose the correct answer. Question 8. Alignment of the chromosomes at the equator of the spindle is characteristic of. Choose the correct answer. Question 9. If a cell with 46 chromosomes undergoes mitosis, the daughter cells will have blank chromosomes. Choose the correct answer. Question 10. You can tell the difference between a plant cell and an animal cell undergoing cytokinesis because. Choose the correct answer. And this is question 10. This concludes our discussion of cell division in prokaryotic and eukaryotic cells. Now we will switch gears and talk a little bit about how this process is regulated in eukaryotic cells and how, when this process is not regulated correctly, you can end up with cancer cells.